Hello, 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 everyone. To those of you guys who were with me yesterday for the practice test or 30 minutes ago for the practice test, welcome back. I'm really, really happy to have you guys here. So give me a shout out if you got, a beverage, you got some food, did some jumping jacks in the interim between our practice test and now. Because if so, you are killing the game. For those of you guys who didn't, you should know that I am very, very pro physical activity during MCAT practice test and test breaks. I'm all about getting up, moving around, getting that blood flowing, getting that fluid flowing, and you guys are going to be learning all about fluid flow today, so I hope you're all pumped. Just a couple of introductions to the Kaplan verse. My name is Ashwarya, and I am your host for today. And I'll also be passing it off to your kidney instructor, John Linnick, who is fabulous. And I'm hoping that you guys are really able to filter through the information he gives you and absorb a lot of that information. I also hope that you guys have fun today. That's our goal. I've seen a lot of you guys posting into the chat, and I want you guys to keep that up, okay? I want you guys to be super social, to talk to me, to talk to John, because that's really how you're going to get the most out of this class, by staying alert, by staying active, by staying engaged. All right, so in the Kaplan universe, a 6 is the equivalent of a thumbs up, and a 9 is the equivalent of a thumbs down. So I want you guys to give me a 6 in the chat if you are ready, you are excited, and you are just super, super pumped to learn about the kidney. So with that, I'm going to jump us in to our agenda for today. So we're going to start out with a welcome and introduction. So congratulations, you guys have made it through step 1. I welcome you, and I, Ashwarya, your host for the evening, am really, really excited to talk with you guys about the MCAT, about Kaplan, about med school, whatever you guys want to know. So I personally am a second-year medical student. I attend Yale School of Medicine, and I'm just loving med school. So definitely reach out, ask me questions that you have about stuff like that. Oh my God, Rachel, I'm so happy you had a dance party. Yes, get that blood pumping, get in that, like, I'm ready to work. MCAT is not as bad. It's actually kind of fun once you get into it mentality. So after we do that, we're going to talk about the MCAT a little bit, just go through what it is, how it's scored, what the subject breakdowns are for those of you who are new, or even for those of you who have been prepping. So it's always good just to see if there's something you didn't know before, just to see it all laid out on paper. Then I'm going to pass it over to John, and he's going to talk to you guys about the kidney. And then we're going to do something called plus coaching. So we have something called plus that comes with some of our courses. And I want to give you guys an understanding of what that entails, what's involved with that. And so that's kind of the part of our session where if you have questions about prepping, if you have questions about applying, I want you guys to ask me all of them. I'm here to help you through this. I know applying to med school can be really, really rough. I was there. It was rough. So I'm basically here to help kind of smoothen that path out for you guys, to help you in any way that I can. So jumping into the MCAT rundown. The MCAT is broken down into 4 sections, but saying 4 sections can be kind of misleading because those sections cover a lot of topics. So we have our first section being Chem Phys, chemistry and physics. Our second one is critical analysis and reasoning, which is the one you guys probably know as the reading section. We also have bio and biochem, and then finally you end your test day with psych soc. Yep, there are 4 big sections on the MCAT. And you'll notice that the critical analysis and reasoning, or we like to call CARS session, because let's be honest, doctors love their acronyms. You'll see that that's a little bit shorter than the other sessions. And that's partially because while the CARS session is all passage-based, your other science sections are a mixture of passage-based questions and discrete questions. So passage-based questions, like their name sounds, require the passage to answer, while discrete questions are really based on your pre-existing content knowledge. So it's really important both to have that content foundation, but also to understand how to take information you already know and apply it to what the test is telling you via the passages. And so that's definitely something that John is going to be talking to you more about as we continue. So are you guys excited? Are you pumped for this session? Are you pumped to learn more about the MCAT and the kidneys? I'm also really excited because I like using puns, and pumped is like a hardcore pun for me right now, especially kidney one. So stay tuned because there will be more. Cool. So I know a bunch of you guys are with me for the practice test. For those of you guys who haven't taken a practice test or taken any kind of diagnostic, I 100% recommend it. Definitely go check out Kaplan events and sign up for the next available practice test because that is the best way for you to figure out where you are and what you should be doing prep-wise to help you improve your score. So those 4 sections that we talked about are each scored independently, and the score ranges from 118 to 132. And when you look at your cumulative score, you'll see that a 500 falls at about the 50th percentile. And so why does this matter? What does this mean to you? Basically, when you take a bio test or a physics test, your score is a reflection of that whole test. And it's a little bit different here because even if you kill bio, even if you kill psych-soc, you want to make sure that you're doing equally well on those other 2 sections or all 4 sections because all of those are reflected in your final score. And so you don't just want to be getting a 130 in Chem Phys, but getting a 120 in the other 3 sections because med schools aren't going to notice that. They're not going to see that score breakdown right away. So they're not going to know that biochem is your jam. All they're going to see is that low score. So like Tim is saying, you want to be a generalist on the MCAT. Everything counts. Keep that in mind as you're prepping. So make sure that, like Tim is saying, even if you're not super gung-ho about psych-soc or biochem or whatever, if you notice after your practice test that that is an area where you could be seeing a lot of improvement, you're dedicating your time to that because you can see a lot of point gains in the beginning if you're jumping maybe from a 120 to a 125. It's a lot easier to make that kind of jump than it is to necessarily go from a 130 to 131, even though those gains are 100% doable. All right, so moving on. What do you guys notice when you see this breakdown of the science content on the MCAT? Because I look at it, and the thing I'm seeing is that Chem Phys isn't just chemistry and physics. Just like biochem isn't just bio and biochem. There's some organic chem in there. There's some gen chem in there. We have a lot of interplay between our different subjects, right? And so that's something that, again, I want you guys to keep in mind as you're prepping. This should always be at the forefront of your mind, that the Chem Phys section is not just going to test you on chemistry and physics. The Chem Phys section is named that because they're going to take concepts that you have learned in chemistry and physics, and they're going to throw them into these whole new contexts. And they're going to see whether you can take those things that you've learned and apply them in ways that you've never have before. And that's why it's important to treat the MCAT as a new beast, to figure out ways of studying for the MCAT that work for you, that are probably not going to be the same as what you used to study for past tests. This is not the type of test where you can just learn, memorize, and regurgitate. This is not that kind of test at all. It's a whole different beast. This test requires a lot of strategy, a lot of analysis in terms of figuring out how you want to approach passages and questions, how they are trying to trick you on different questions and things like that. And so again, I'm really excited for you guys to spend some time with John because he'll be touching on all of this and he'll be giving you a first-hand view on what I mean. And so this is, again, just throwing it out there. The MCAT is not just a test that tests science knowledge. You are going to be tested on your scientific reasoning. So do you understand how to set up an experiment? Do you understand what experiments are testing? Can you read those results of those experiments? Can you take the skills that you are going to need in med school? The ability to sort through all this information that a patient is giving you in order to pull out the relevant stuff. Are you able to incorporate, someone's medication history with their past surgical history and use that to guide your diagnosis of what they might be presenting with now? It's all really similar in terms of logical thinking style to what they're testing on the MCAT, which is why it's so important to make sure that you are building these skills up now so that when you get to medical school, you already have a wonderful foundation. You already know what you're doing, and you're just it's a matter of taking in new information and applying it into the system you already have. Cool. And so what we're doing today is going through an MCAT channel session. So an MCAT channel session is a way for you to basically focus on the information that maybe you don't know as well, or maybe after you've taken a few practice tests, have noticed that even with content review via a book, the information is just not sticking. And so what you're going to notice as we go through is that John is going to present information to you in a very MCAT specific way while still conveying the high yield points of that content. So keep that in mind. Definitely go ahead and take notes, and also pay attention to the skills he's going to teach you, because those are things that you can use in any section, not just this one, not just with respect to the kidney. And before I hand it over to him, I also want to draw your attention to the top of the chat, and you'll see that there is a link pinned there that says Kaplan-Dogs MCAT Prep for the Win. So if you haven't clicked on it yet, I want you to go ahead and click on it and fill out the form, because that'll enroll you in the raffle to win a Starbucks gift card for $50. And we'll be doing that raffle at the end of the session. So definitely go out and fill that out now. We've had 2 winners already in our last 2 practice tests, and it's definitely a fun way to get involved. And with that, I am going to pass it over to John. Like this slide says, your journey to a higher MCAT score begins now. So I'm really, really excited for you to meet him. He is one of the people that I personally watched when I was prepping for the MCAT. He too is a former Kaplan student. He's an avid chai drinker, and he is coming to you now.
Hey everybody, thanks for the intro, Aishwarya. She and I sometimes talk about to drink dirty chais, and so it's funny that you said that. But welcome, it's nice to see you here. We've got a lot of fun stuff to talk about, and I'm going to try to continue with the same thing that Aishwarya talked about, and that's how all of the sections on the MCAT, well, not all, most of the sections on the MCAT test the same ideas, just in different contexts. And so this, of course, we're going to cover the kidney. Yes, it's physiology. Any guesses what section of the MCAT you would most likely see kidney questions on? Because remember, it's not necessarily just one, but you will see heavier amounts of these kinds of questions in some topics other than other ones. And so, as everybody expected, this is really common in the biochem section. You get a lot of the physiology and the bio there, exactly true. Saying. But you could also see this in the Chem Phys section. So like here's an idea of how this could also get tested in Chem Phys. One of the main ways that the kidney functions is with different concentration gradients. So it's dealing with osmotic versus oncotic pressure versus hydrostatic pressure, which is why you can actually see it in Chem Phys as well as Laura and other folks are saying too. And so yeah, lots of bio, but beware, don't study certain topics on the MCAT and think of this as, oh, this is a bio topic. It's not. It's all connected. And the more that you start to draw those lines together, the easier it'll be to really make sure you've got all your ducks in a row in test day and can rock this. All right, so we'll jump right in. And so we're going to go through a lot of different things. Out of curiosity, though, who's studied kidney physiology before, though? This is one of the parts of the MCAT, or one of the sections of physiology that is really, really, really important for the MCAT, partially because of the reasons we talked about before, but it also connects to things like endocrinology. It connects to, of course, like whole systems approach. You can see lots of kidney stuff, and I would say kidney and circulation are some of the highest yield topics that you definitely need to know for test day. And so make sure if you haven't, nice, lots of folks study it, perfect. And so if you haven't studied it before, make sure you're comfortable with this organ specifically. And so we're going to do it today. It's why we're doing it in today's class, because it is so important. Nice. And yeah, so Nadia's bringing up a great point too about in anatomy classes. So one thing as a heads up, you don't need to know anatomy for the MCAT. It's actually not tested. But if you've taken anatomy classes before, or if you've done things like working for a nephrologist there, and awesome, you can use your background knowledge to apply to the context that you do need for the MCAT. And one of the big ways that you'll do that is word roots. If you know your word roots, you can often figure out so much more. Let me give you one example. So what's the name of the organ that's directly above the kidney? This is going to connect more to endocrinology. And so it's not necessarily directly just kidney physiology itself, but I could even draw a little picture of it right up here. So I like to draw it as a little hat that hangs out on the top of the kidney there. And so you guys got it. So the organ on top of it is the adrenal gland. Good call. And so the adrenal gland, notice what it's called. Adrenal. Ad is an adjacent, renal. Notice the renal system, that is your kidneys, right? Exactly as Tim's breaking it down there. I mean, going even further, one of the organs that's released from the adrenals is, of course, adrenaline, right? Released from the adrenals. But the proper name for adrenaline, at least in the States, I won't get into the history of copywriting terms, but so in the States we call it epinephrine. Think of the same word roots there. Epi, like epidermis means on top of, nephrin, nephrons. Where do you find nephrons? Huh. Well, it's what we're going to talk about here. It's in your kidney, of course. And so the cool thing is if you get really good at your word roots, you can figure out all of this stuff. And so, well, not all of it, but a lot of it. And so we really want you to focus on the big picture things, how everything connects, and word roots are a great way to do that. All right. So let's talk about a couple major functions of the kidney. We did already talk about, well, I guess we'll talk about the nephrons in a second here. But so they do lots of things like regulating pH, blood volume, osmolarity, get rid of waste products. One of those main ones is urea. That's a byproduct of eating protein. And so if you eat a lot of meat, you tend to have more urea in your urine, where it comes from. And that's one of the things we excrete, as well as lots of other stuff as well. So remember, though, that when anything that you're excreting through the kidneys, it's pulled from the blood. And so you filter in your kidney, we secrete, we excrete, we'll go through all the different functions that we do here, but it's really just filtering your blood and taking all the stuff out of it. So when we're talking about this, one of the major units that we're going to use is osmoles. Does anyone happen to know what the common osmolarity of your body is? In osmoles, there's different units you can see if you work in hospitals, but we're going to try to focus this on ways that you see it on the MCAT as much as possible. And so if an osmole is 1 mole of solid particles, that means that you don't care about what those particles are, just how many particles there are. And so Hassan's right, it is 300, but the units there, it's 300 milliosmoles per liter. You're totally right, it is 300. So yeah, 300 milliosmoles per liter. So that's the standard concentration in your blood, that's in your cells, that's in your lymph. That's just the standard concentration in a human in general. And so now the kidney actually has the ability to concentrate that even more, and we'll go through that full pathway, and we'll show you the full loop of Henle and the entire process. It should be pretty fun. All right, so now we already talked about the adrenals, and that's your little hat that I'll put on top of there. So the adrenal gland is the top of the kidney. One more common word root that I want to point out here. Notice that the cortex is the outer layer of the kidney. The medulla is the inner layer of the kidney. Can you think of any other organs that have an outer layer that's a cortex? But then if you look at the inner layer, it tends to have different physiological functions, and it's known as the medulla. Kidney is also specific. It has a pelvis beyond that, but the common word roots here are cortex and medulla. You do see that in things way more than just the kidney. It actually shows up in a bunch of different organs. One of the more common ones that people talk about, of course, is our brain. You've got it, yeah. So brain, other organs as well do it. You can even talk about your adrenal. Actually has an adrenal cortex as well as adrenal medulla as well. It releases different hormones depending on where you are. But so this is a common word root. You can see it both from lymph nodes, the brains, the kidneys, all over the place. So word roots. We'll keep coming back to word roots as much as we can. All right. So in terms of hormones, there's a couple hormones that are important. The first one is erythropoietin. Once again, word roots, erythro, like erythrocytes, red blood cells. Poietin, potential, so that gives you the ability to create new red blood cells. That's EPO. In terms of pop culture, this was actually the hormone that was abused by lots of professional cyclists. They were basically taking erythropoietin to thicken up their blood, to add more red blood cells so you can basically bicycle faster or longer. Of course, you can also do this naturally without taking hormones by just training at altitude. And so if you go up to high altitude places where there's thinner air, your body naturally creates more red blood cells to deal with it. That's actually why the United States Olympic Training Institute, I think it's called an institute, but where all the Olympians train is actually in Colorado Springs at about 7,000 feet or so. It kind of naturally gives people more red blood cells, and it isn't breaking the rules when you do so. Exactly as experts. You see, Tim and I, we're thinking the same thing. Cool. All right, so there's erythropoietin, calcitrol. This one you might not know, but it's actually an active form of vitamin D, so I'm just going to put vitamin D real quick. You've probably noticed this before, but if you ever, well, not if you ever, but if you see orange juice that they add calcium, they'll often add vitamin D as well. Or you've probably seen vitamin D milk. There is a direct relationship between vitamin D and calcium. It is an important relationship. We cover it really well in depth in one of our MCAT channel classes on endocrinology. But for now, word roots, calcium, vitamin D have that association down. But we do get specific with that on the MCAT channel, which I teach a lot. It's really fun. All right, a couple other things. Synthesis of glucose from amino acids. Anyone know the other organ that you can synthesize glucose from scratch? It's kidneys, not the most common one. Actually, people usually don't think of the kidney when you think of gluconeogenesis. Once again, word roots glucose, neo, new, genesis to create, kind of like Genesis in the Bible, right? You know, the creation story is there. It's all the same word roots. But so yeah, if you see it's mostly liver, exactly, Chris, and everybody's saying you got it. So if you see gluconeogenesis, first think liver. That's the more important organ that does it. But kidneys actually do have the ability to do so as well. Cool. And oh my gosh, we get really into all that stuff on the MCAT channel. We have a lot of metabolism classes that go through the entire metabolic pathway from, you know, I guess all the way from glucose all the way down to energy and beyond, and fats too. It's fun. But for now, though, we're going to get more into not just kidney, but the idea of concentration differences, pressure, and the idea of how this fits in a big picture. So notice this is your cell membrane. So imagine this is a big cell. If that's a capillary membrane, that means this is your blood vessel here. So I'll put like blood right there. But there's actually a space between the blood vessels and the cells known as the interstitium. Now, both the blood, which is your plasma, right? That's the liquid component of your blood, and the interstitium are known as extracellular fluid, as you can see there. Inside the cell, of course, that's intracellular fluid. Now, one thing to point out, a lot of people don't really think about this, but when you think the relationship between your capillaries there and the interstitium, capillaries actually have a lot of holes in them. They're known as fenestrations, to use the proper word there. But the thing is, it's important to let things go in and out freely. Because if you've got a lot of oxygen, if you've got carbon dioxide you want to get rid of, if you've got sugar, that stuff needs to make it into the cell. And you don't want to have to actively transport it out of the blood, into the interstitium, and then again into the cell. And so you can actually freely move things between the plasma, which is the blood, and the interstitium, unless what? So what kind of stuff can't just like escape the plasma and make its way into the interstitium? Because we said small stuff like oxygen can, even glucose can do this, and vitamins can do this, gases like oxygen, carbon dioxide, all those are perfectly fine to do so. But certain things can't. And it's going to be one of the most important parts of today's class when we talk about concentration differences. Exactly, a bunch of folks are saying it. Albumin, big stuff. Now, ions actually can. So like sodium, chlorine, ions, they're fine because you've got fenestrations in this capillary bed. And if you've got a hole, even if it's charged, it can move through. Now, generally speaking, ions cannot make it into cell membranes because a solid phospholipid bilayer, ions can't make it through. Capillaries are different, though. The only thing that can't make it through is big stuff, and as you can see, a bunch of folks have already said in the chat, that's proteins. Namely, I like how someone brought up the specific one at the very beginning. Laura has mentioned it, albumin. Good call. We're going to talk a lot about albumin in this class. It is the most common blood protein, and it's going to make a very big difference for all these conversations. So notice sodium, ions fine, they can freely move back and forth, but albumin is going to be way, way, way higher in the plasma, because it's your most common blood protein, and it's so big it can't move back and forth. So now there's two main things that you really want to know about albumin for the MCAT. Of course, there's way more, but we'll do the two big things for the MCAT. Is that it establishes the oncotic gradient. So I want to actually draw this real quick. So we have oncotic versus osmotic. All right, so oncotic versus osmotic. These two are the same general idea. So let me give an example. So let's say that you have a saltwater fish. All right, here's your question, everybody. So let's say you've got a saltwater fish. We can name him Nemo, maybe. And we'll say we got the saltwater fish named Nemo decides to jump into a freshwater toilet. Not a good idea. But what would happen if you've got this super salty fish that all of a sudden is exposed to this hypotonic environment? And to make Nemo look even saltier, I'm going to put a bunch of red dots all over him. So there's all the salt. Once he jumps into the toilet, though, the toilet does not have as much salt. Thus, it's a hypotonic environment. Right, Sharia? What would happen? Yeah, Julianne. Why? Why, everybody? Why would he actually explode? Because it's true, Nemo would probably explode. So here's the thing, Jose. It's not that he could lose salt, because if you think of his own cells' membranes, ions can't flow back and forth between cells, membranes, but water can. And so what's going to happen is that water is going to just flood into this poor little fish, which will cause him to swell and eventually explode. And because I like to use this Nemo example a lot, I actually did some research, and it turns out Nemo could have survived if he was flushed fairly quickly. You can put a saltwater fish in fresh water for, I've heard it's like up to half hour or so, and they usually won't die yet. And so it is actually possible. Nemo could have made it, but in general, if very salty things go into fresh water, as you guys are all saying, water is going to move into its cells and cause them to rupture and burst. You got it. So that is osmotic pressure, salt, ions, that kind of stuff. When we talk about oncotic pressure, though, oncotic pressure is the same thing, but it's that concentration difference that's due to proteins. Big stuff, to be more specific. It's like anytime you got really big stuff that can't leak out, then we refer to it as oncotic pressure. If it's just salt, things that can move through fenestrations, then it's just the more general osmotic pressure. And so they do mean the same thing. I just want to be really specific here, because when you talk about the kidney and blood vessels, it is oncotic pressure, because as you can see here, that sodium can actually freely move back and forth. So you need proteins that actually make a big difference. Cool. All right. So there's a little background, and of course a little connection to Nemo as well, which is always fun to connect whenever we can. All right, and so let's talk about a couple other things here. So we've talked about albumin. We mentioned lymph nodes briefly, but we didn't talk about the lymphatic system too much. And I'm actually going to erase this Nemo picture here just to make some space. But so what the lymphatic system does, it's like your circulatory system, but it doesn't have a heart. And so what happens is, since all of this fluid and salt and stuff can just leak out of your capillary membrane all the time, this interstitium needs to get drained once in a while. And that's the function of the lymphatic system. It basically picks up that extra fluid and dumps it back into the blood. It's a very roundabout way. It actually does it. It's called the thoracic duct. It's up in your chest, near your heart. But what happens is that all of these lymph vessels basically go next to all of your blood vessels, all of your body. But since they don't have a heart, do you guys actually know, like, how does your lymph move? Because your blood's easy, because you got a heart to pump it. But since the lymph doesn't have a heart to move it, something else needs to, like, force that lymph to actually migrate through your body. Oh, great question, Laura. I'm not sure about oncotics word roots. That's a great question. I've not looked that one up. I bet one of the other teachers might know that. But do you guys know how you get your lymph to actually start flowing? It's really important when you think about people getting sick, because your lymph system is highly associated with your immune system. Oh, nice. Thank you, Ashwarya. Exactly. Perfect. It doesn't have valves like vein. Well, that's not so that valves wouldn't get it to flow. They would stop movement. You want it to actually keep moving, though. But exactly, Ganche, muscle contraction. Exactly true. And so the thing is, unless you're actually contracting your muscles, or getting massages actually works fine too, your lymph does not move. The reason this is a big deal is because you need your lymph system, because it's where a lot of your white blood cells live. And if lymph isn't moving through it, your white blood cells aren't getting exposed to the bacteria that they need to fight. And so if you're sick and you stay in bed and you do not move and don't contract your muscles, that means your lymph's stagnant and you're not going to get healthy as soon, and you also get some swelling. So it's really important when you get sick to still get up, to move around, try to walk, or if you're really lucky, just get some massages, which would be really cool. But you got to make sure that lymph keeps moving. It's very, very important. And you'll see a lot of hospitals will do this. They'll actually get patients that are bedridden up. They'll put them in ambulatory devices and things like that. And I've actually done some volunteer work in hospitals, like helping with that kind of stuff. It's really, the sick need help. We got to help them out. That's why you're all here in the first place. Right? All So there's your lymphatic system. A couple of things I want to point out before we move along, and that is the differences between all of these concentrations you can see in the bottom corner of your page. This is a dense page. All right, so notice that you have high sodium extracellular, high potassium intracellular. That's due to this pump right here, which is the most common transporter on the MCAT. It is your sodium potassium ATPase. You need to know this protein transporter. All right, it's a protein that transports ions, right? It doesn't transport proteins. It is a protein. It moves ions back and forth. And so notice you pump 3 sodium out, 2 potassium in. Another Kaplan teacher taught me this mnemonic. I can't remember who it was right now. But the way that they always remember it is to think of it as a bouncer. So notice sodium. Well, if you're a bouncer, you want sodium to come in. Nah. Notice it's getting kicked out. Nah, not allowed, right? Na, sodium. Whereas the potassium, okay, you can come in. And so it's kind of like your cell's bouncer, and it's telling the sodium's no, and the potassium's okay. I don't know, it's cheesy, but you do got to know that for the MCAT. It's really important. All right. So let's do one more. Phosphate and inorganic ions. Why in the world would you expect to have lots of phosphates inside of your cells? This is actually one of the really common polyatomic ions that you'll see all over the place. And also as a little bit of a background, it's notice it says phosphate and organic. That's because phosphate's inorganic. Anything that doesn't have carbon will be considered inorganic. Sorry, the sun's setting, so the lighting's getting a little weird here for a minute. But nice. You got it. Totally true. ATP. And so we're constantly making ATP. You've got tons of enzymes known as kinases that are constantly phosphorylating things. But oh my gosh, phosphate has a lot of jobs. Not just ATP, but think about your DNA in general. The backbone is a sugar phosphate backbone. Of course, all of your kinases and metabolism, phosphorylation, cyclic AMP like CAMP. Oh my gosh, there's so many places that you see phosphate used over and over again. So it's one of those ubiquitous molecules you'll really see everywhere. Cool. All right. So there's a bunch of stuff here. I'm just kind of introducing the different things that are floating around. What we want to do for the rest of class is to dig into the kidneys a bit more specifically. And one thing that we did already mention, I'm going to erase all my scribbles here, was that, remember, your body is 300 milliosmoles per liter. It doesn't matter if it's the blood inside the cells or the interstitium. That is just our body's concentration. That's what we like to be. All right, so let's start with the questions. So actually, Aishwarya, I think you've got the polls. If you want to grab a poll, we're going to actually have you all vote for question number 1. Let us know what you think is going to happen, and then we will chat about the results once they come through. Awesome. Thanks, Aishwarya. Cool. So keep the votes coming in. We got most of them in. Looking at the results right now. Cool. Sweet. All right. So I'm just going to talk this through. For those of you that haven't voted yet, please get your answers in. But so if we have a sharp rise in the serum albumin, that means blood albumin, right? So that's the liquid component of your blood. And so if I were to draw a little picture here, this basically means that now your blood vessels have suddenly gotten much more concentrated. And so if your blood vessels suddenly get way more concentrated, that means, since this is albumin, you have increased the oncotic pressure of the blood. And there's a kind of a cheesy mnemonic, but whether you're talking osmotic pressure or oncotic pressure, it sucks. Because that's what it does. It sucks fluid into your high oncotic pressure. And so water will get drawn into the bloodstream. And that water, of course, the bordering environment is, as 50% have said, the interstitium. So yeah, you'll have an influx of interstitium fluid into the bloodstream. Nice work. All right, so let's get into physiology. As we said, we're going to end with physiology here. We're going to go through all of the different structures of the nephron, what they do. But the first and most important thing to mention is the functions of the kidney. And so we mentioned these before. Filtration. So filtration occurs at the glomerulus. That's this part right here that I just circled in blue. It's a highly fenestrated, notice this word keeps coming up, capillary bed. So it's got lots of holes in it, which means small stuff makes it through, big stuff does not. So that's the key. Notice no proteins, no cells make it into the nephron. They stay in the blood where they belong. So that's filtration. Glomerulus. That's where it happens. So it filters out the small stuff, makes it in. So now all that liquid, well, not all, but a lot of that liquid just made it into your kidney. It makes it through this Bowman's capsule. This is the next part. We're not going to talk about that too much. Into the next important region, which is the, as you can see, proximal convoluted tubule. So what's going to be important? If you just got all of the liquid in your blood, not the cells, to get basically into the kidney, what do you want to make sure that you save? Because notice, if you make it all the way through the nephron, it's getting into the pelvis, into the ureters, and you're going to pee it out. So what kind of stuff do you want to make sure you definitely do not pee out, because it's going to be a waste of time? Oh, and Alberta, I just noticed what you chatted. You're totally correct. Yeah, influx of interstitial fluid means an increase in blood pressure. Sorry, I missed that. I didn't notice you said that. But yeah, totally correct. More fluid in the blood means more blood pressure. Good call. And yeah, you guys are totally right. You want to save the sugar. You want to save a lot of the water. You want to save the useful stuff like calcium, glucose, exactly. Anything that is useful is what you want to save. Vitamins are another big one there. And so that's known as reabsorption. So you want to reabsorb all the good stuff. Glucose, amino acids, salt, water, vitamins, all that kind of stuff you want to save. And so notice when you say reabsorption, that means taking it from the nephron back into the blood. So that's what reabsorption means, taking it from the nephron and back into the blood. Sorry, I bet that yellow is a little hard to see. I'm going to change the color to a nice Kaplan purple. There we go. Cool. All right, that's reabsorption. Also, you want to have the ability not just to have stuff getting filtered out through the glomerulus, but to actively get rid of bad stuff. And do you guys know what the stuff you want to make sure you get rid of is? There are certain things that you just definitely do not want to keep because you want to get rid of them. They're waste products. They're floating around in your blood and they can cause problems. And so this is the stuff that you definitely want to get rid of. And this is known as secretion. So secretion means you're going to actively take it out of the blood, and I'll label this as well. Taking it out of the blood and putting it into the nephron. And remember, stuff that ends up in the nephron eventually gets excreted. Excreted means leaves the body. And once again, I seem to be auto-prompting to this yellow font, so I'll change it to purple once again. All right. And yeah, as you guys are saying, urea is a big one. Good call. So urea, protons. When you get acidic, you want to get rid of extra protons. Actually, potassium is another common one. The potassium pathway is kind of complicated. I was talking to some friends that are studying that stuff, and they said, don't even talk about it to pre-med students because it's beyond the scope of what we care about. But potassium is common. We'll leave it at that. The balance is apparently over the top, though. So there we go. So that's secretion, which notice also we've previously said in the chat a second ago, as Tim mentioned, things that get excreted means it's exiting the body. So here's a little formula just to show how that works. So excreted, what leaves the body. So that's all the stuff that was filtered, thus made it into the nephron. Then subtract the stuff you wanted to save, right? That's the good stuff you reabsorb, put it back into the blood. And then just add in the extra stuff that you want to get rid of. That's the secretion at the very end. All right, so that is the function of the kidney, what it's really doing. And we'll talk next, well, not immediately next, but soon, about the various regions like proximal versus distal versus loop of Henle, and show you a cool animation that really relates it all together. And of course, here's the actual anatomy of it, right? It goes from the kidneys to the ureters. Ureters lead to the bladder. Bladder leads to the urethra, and then urethra exits the body. And this is actually why it's super important if you ever do have a UTI, a urinary tract infection, you gotta take care of it, because if that infection travels all the way up to the bladder, that means that now you can actually get it to go all the way up to the kidneys. And if you get a kidney infection, you are not leaving the hospital for days. It's a very, 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 very serious infection. And so it's really important to make sure you take care of them as soon as you can if they do ever happen. All right, so this is just a recap of the stuff we already talked about a little bit before. Yeah, seriously, right, Tim? And so this is, notice as we said before, filtration occurs at the glomerulus. Now, this isn't saying that's specific to that's the loop of Henle. It's just that secretion means from blood into nephron. Reabsorption means from nephron into blood. So this is actually just a recap of the same stuff we talked about, just showing an image. But be aware, this is not location specific. It doesn't happen right from the blood to the loop of Henle. It's just showing you what it's trying to say there. But yeah, it's not location specific. All right, so let's just recap. We saw that before. All right, so now we're going to get into oncotic pressure for a little bit here. This will be a little dense, but I think it'll be fun. So notice we have a couple different types of pressure here. This pi symbol stands for oncotic pressure. The P stands for hydrostatic pressure. So hydrostatic pressure is like pressure in a garden hose. If you put your thumb over it, it sprays out. That's increasing the hydrostatic pressure. Or you can think of altitude differences. You see hydrostatic pressure in physics a lot. But so the thing is, you have a balance of two different types of pressure. You've got your heart pumping, which is forcing fluid out of the blood and into wherever it can get to. So that's the hydrostatic pressure of your GC, stands for glomerular capillary. More importantly, just the capillary. And so your blood's actively squishing fluid out, right? Whereas your blood's also actively pulling fluid back in due to the blood's own oncotic pressure. And so in terms of the balance here, the hydrostatic pressure of the Bowman's capsule is pretty darn negligible. There's not a heart pumping it there. Same thing for the oncotic pressure. It's really not that strong. So the real big balance here, what matters most, is you've got your heart pumping creating hydrostatic pressure, though since the blood also is concentrated, it pulls fluid back in. The other two, of course, are there. Yes, they do make a difference, but comparatively to the other stuff, it is not that important, which is why we're kind of going through all this stuff. Cool. All right. All right, so this is known as Starling forces. I'll give you the formula. Actually, I don't know if we do. Do we have the formula here? We might not. I think it's at the bottom page. There it is. So So this is known as Starling's law or Starling forces. This is really just what we were talking about before. Since you have such great that's hydrostatic pressure in the glomerular capillaries, right? Since your blood has really big hydrostatic pressure, it forces fluid out of the blood and into the nephron. Yet, since the oncotic pressure of the blood is so great, it's going to cause fluid to flow back into the blood, thus out of the nephron. And so this creates a balance, yet there is still net flow from the blood to the nephron, because notice how much greater that hydrostatic pressure is there. So you don't need to memorize this formula here. It's really just to get you all to think about the balance here. Exactly as Tim's saying in the chat, like we have a balance between all these different forces. What's most important is that fluid leaves the blood, goes into the nephron, because that's where it's going to get filtered and treated and processed. Cool. All right, so that's a little introduction. If you want to nerd out on this, feel free to do a little bit of searching. We talk about a lot in our, I guess, a little bit in our courses and a lot in our books. But so it's mostly just having the general idea down that's most important for the MCAT. All right. So let's do a couple questions here. So notice you have, once again, important word roots. Your afferent arteriole. And what does an afferent arteriole mean? Does that mean going inwards or is that going outwards? So is it entering or is it exiting? And you'll actually see this exact same word root with neurons. And I'll give you the neuron breakdown here too. But so your there's actually a mnemonic. It's same, so it's sensory, efferent, motor, efferent. So this is actually for nerves. Nerves, exclamation point there. But nice, as you guys are saying, it's definitely entering. Good call. So it's the same thing when you think of sensory, right? Sensory information is coming into you. Motor is exiting, that's going out of you. And so if you look at this nephron here, your afferent arteriole means it is entering the kidney. Your efferent arteriole means it is exiting the kidney. So notice efferent exit. It's kind of a nice way to remember that. All right, so now what we want to talk about, though, is what would happen if you were to constrict, well, how about this? What would happen to the glomerular filtration rate? That is known as your GFR. You'll see that abbreviation quite a bit. So GFR, glomerular filtration rate, if you were to constrict the afferent arteriole. So notice, if you're going to basically block blood from making it into the glomerulus, then you're not going to have as much GFR because you're just going to stop it from getting in. It's going to cause the GFR to thus decrease. You got it. And then the exact opposite would happen if you were to constrict the efferent arteriole. If you constrict the efferent arteriole, it's going to cause a backflow. All this fluid's going to enter here, have nowhere to go, and so it's going to have no choice but to start to squirt out through the glomerulus, which is why you have an increase in GFR. Now, last one, what happens if you get a kidney stone stuck in your ureter? So that's way downstream, right? So what do you guys think would happen to the GFR? And I'll try to draw this out as you're thinking about it. But so if you were to extend this all the way down to the kidney, I'm just going to say like this is an approximation of the entire nephron. All the way here, you got your ureter. Sorry, I'll be a little sloppy there. But so if we were to basically plug the very end of the pathway, that means you're not going to get any fluid to leave the kidney. And if you don't get any fluid to leave the kidney, it's, first of all, as Nicole's saying, going to hurt really, really, really badly. But then you're also not going to get any blood, or any, I guess technically it's known as a filtrate, to actually exit the kidney either, which means blood can no longer enter the glomerulus as that pressure builds up, which would cause an actual decrease. Gotcha, saying totally true. It actually causes it to go down. This is very, 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 very scary, because if you're going to stop blood from leaving your kidneys, that means you're no longer able to filter your blood. And do you guys know what medical treatment you give someone who loses their ability for their kidney to filter their own blood? If you have kidney failure or your kidneys aren't functioning, you need to get this, or it will kill you fairly quickly, within a couple days. That's why kidney infections are not something to mess with. Yeah, exactly. Light really sits me really bad. Back up. Dialysis, yeah. So that's what dialysis is. Dialysis is basically plugging. Depends how the patient's getting it, if it's long-term or not. Sometimes they'll put a port in. But basically, you take the blood out of the person, filter it through a machine like a kidney would, and then put it back into the person. It takes several hours. I imagine a lot of you have had loved ones or friends or have worked in hospitals where it's been going on. But it's a big part of someone's life when they have kidney failure. It's usually 3 times a week they'll have to be going to get dialysis. It's really significant. But it can keep you alive for a very long time, where otherwise people would die. That's pretty awesome. All right, so that's a little bit of filtration. So we'll just go through this kind of quickly. We'll rock this out and jump to our last part of class. But this is what we mentioned before. Cells and platelets should not make it in, right? They're too big. They can't make it through. Ions, sure. Glucose, sure. But big stuff like proteins and cells and platelets don't. And so that's the only thing that does not make it in, is the big stuff. Small stuff does. Remember, this is different than cell membranes, right? This is a fenestrated capillary bed, and even ions can get leaked out. It's just the big stuff that can't. All right, so let's get into we'll kind of get into the different sections here. But so notice the first one we have here is secretion. As we mentioned before, secretion is when you're taking stuff out of the blood, you're putting it into the filtrate. So this is, as we said before, it's extra protons, potassium, urea, nitrogen, that kind of stuff. And also, for those of you that don't know the structure of urea, urea is like it's kind of like acetone for amides. And so notice it's like acetone, but you actually have a nitrogen on either side of it too. And I'll draw it unprotonated. But yeah. So that is urea. You do want to know that structure is a common waste product. Just like acetone, but it's nitrogens instead. Thus, it is an amide. Very common word root there. All right, so that's secretion. Notice that it happens at the tubules, both proximal and distal convoluted tubules. So why do you think these are so convoluted? Right? Sometimes they're complicated. Like, if we were to draw them, that means that they're really twisty and turvy and bendy and whirly all over the place. So what's the benefit of that? Like, why would you want these tubes to be so twisty, turvy, and convoluted like that? Because it's not just kidneys. You'll see this all over the place in lots of different physiological systems. Actually, the mitochondria has a similar setup. Exactly, Anastasia, you got it. It increases the surface area. Good call. The more surface area you have, the more transport you can have. You'll see that for mitochondria. You'll see that in your brain. You'll see that in your lungs with all the alveoli. You'll see it with the villi and microvilli in your intestines. It's all over the place. But exactly, you always want to maximize surface area. Good call, everybody. Cool, so that's your convoluted tubules. And notice this is where we're secreting as well as reabsorbing. Notice it's primarily the proximal convoluted tubule that you reabsorb all the good stuff. Cool. All right, so this is what we talked about before. Eat a lot of proteins means you're going to have more urea, so you're going to have to pee out more urea. And that's actually the part that makes your urine yellow too. So if you're a veggie, you might not have as stinky of pee, which is kind of nice. All right, and as we said before, anything that doesn't, this is just stuff you want to get rid of, even if it's so big that you can't get rid of it from the glomerulus. That's what secretion is. All right, so reabsorption, we mentioned this before. This is all the good stuff, right? So you want to basically take it from the filtrate, put it back into the blood. This is, once again, primarily at the proximal convoluted tubule. That is the thing you want to note for that. So primarily at the proximal convoluted tubule, but it happens other places too, of course. But that's where the really big stuff happens there. So from the filtrate into the blood. So we'll pretty soon get into the loop of Henle and talk about the physiology there because it's pretty rad, but we'll get to that soon. But notice this is just terminology: reabsorption from filtrate into blood, secretion from blood into filtrate. All right, we actually have another on the MCAT channel. We have an entire class dedicated to these two hormones, ADH and aldosterone. And so it's actually, we teach it generally after this. So we're not, of course, today, but yeah, check it out. It's on the MCAT channel. We have so many cool classes on the channel. It's awesome. All right, so here's a quick little pathway. I just want to talk through this. Well, actually, first, I want to give you all about 20 seconds to figure out what's going on here. Because you might notice whenever you see a graph on the MCAT, your first job is to identify what your axes are. So x-axis is plasma glucose concentration. Y-axis is glucose either filtered, reabsorbed, or excreted. That or tells you that it's three different things. So that means you've got three different lines. Notice you've got blue, you've got red, you've got yellow that relate to either filtered, reabsorbed, or excreted. So I challenge you now, just take about 10 seconds, figure out which line represents which process. And as Aishwarya and Tim were saying, oh my gosh, units, axes, so important. And so, any guesses what this biggest line, this blue line, would be? And notice you can actually tell if you look at these two, if you were to take that red line and this yellow line and add them up, it would actually equal that blue line. Because as soon as the red line plateaus, that's when the yellow line starts to pick pace up. Nice, Anastasia, rocking it. Shelley's saying the same thing. You got it. So notice what's going on here is that the blue line is your filtered load. That's the filtration that's happening. Notice the more plasma glucose you have, the more glucose makes it into your filtrate. The red is the amount that you can reabsorb. Notice that you can only reabsorb to a certain amount. It looks about it looks like it's around, what is that? Somewhere around there. 300 and something, what our units are, milligrams per milliliter, it looks like. But you can only reabsorb so much glucose. That's why people with diabetes, if you've got crazy amounts of sugar, glucose in your blood, you eventually start just peeing it out because you have hit the reabsorption maximum. You've maxed out your transporters, no more can get reabsorbed. And that's how they used to test for diabetes. Docs would just drink people's pee, and if it was sugary, you know that person has diabetes. I actually don't know if that's a myth. That's what they say. I think they would just have people pee outside and see if ants and bugs went to it. Yeah, right, Tim. That's the myth. I'm not sure if it's actually true. All right, and then the yellow is exactly, you call it, Anastasia, that would be your excreted. So notice it's yellow. Funny, just a joke. But that's the part that actually does get peed out. So notice as long as your blood sugar is under 200 or so, you should not be peeing out any glucose. And if you are peeing out glucose, you need to see a doctor immediately because it means you might have diabetes, which needs to get treated. Cool. All right. So now just to mess with these a little bit, notice that the molecular weight was larger. Big molecules don't get into the glomerulus. So that would basically flatten out that filtered load because you have an inverse relationship, right, between molecular weight and how much stuff makes it into the filtrate. So you wouldn't have as much being filtered. And then if TM was larger, that TM stands for transport maximum. That's, at this point, notice the maximum amount of glucose you can reabsorb. So if you had a larger transport maximum, then you wouldn't pee out glucose until your blood sugar got to a really, really, really high rate. So like the yellow curve wouldn't start until later on, is one way to think about that. Cool. All right, so the reason we bring this up here is because often on the MCAT, you're not just going to be tested on the content and the physiology and the biology. It's testing it in the context of graphs or relationships or even physics and other things that you wouldn't expect. And so if you don't feel comfortable with data interpretation and graph interpretation, you need to work on that stuff first. Super, 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 super important. Nice and awesome, Emma. So it is true. I wasn't sure if it was I didn't know if that was a myth or not, but yeah, it was a diagnostic tool. Not surprised. All right, so what we want to end class with today is actually a sweet little animation. So we'll take a couple minutes to do this, but this is our last slide of the day. So this is the countercurrent multiplier system. It's going to give us an opportunity to talk about all the different physiological regions of the nephron. A lot of this we've already brought up, but we didn't get too much into the loop of Henle yet. So this will be our loop of Henle conversation. So as we said before, you've got the horizontal segments, your proximal and distal convoluted tubules, that reabsorb or secrete stuff. The vertical things are what we haven't talked about yet. This is what deals with volume and concentration differences. So now, to get real specific here, what people don't realize all the time going into this is that as you get deeper and deeper into the medulla of the kidney, it gets more and more concentrated. So this is an ugly drawing, but as you can see, I'm drawing more lines just to show you that as you get deeper and deeper down, you are getting more and more concentration. So it's more concentrated down at the bottom there. All right, so as your blood basically gets filtered and becomes filtrate in that proximal convoluted tubule, as it starts to go down the descending loop of Henle, and the environment gets more concentrated, that means that you're increasing the osmotic pressure of the environment, which is going to thus suck water out. So notice at the very beginning, we're going to reabsorb the good stuff. We talked about that before. But as you go down that loop of Henle, notice the filtrate starts to get more and more concentrated. That's why it looks yellow there. It's because the environment is more concentrated, so that's going to draw water out and concentrate it. Now the thing is, this is all fun and good, but as you go back up, what would happen is water would just flow right back in. And you do not want water to flow back in. I'm going to X that out. That should not be happening. So your other option is to actively pump out salt. So this is known as the thick ascending loop of Henle. Notice these little symbols there show active transport. So you're actively transporting sodium out in the ascending loop of Henle. Cool. Awesome. All right. And so this brings you back to your standard concentration. So basically, once you get up to the top, it's the same concentration as before, but notice what the net benefit was that you've reabsorbed, right, out of the filtrate into the blood, a bunch of water, and you've reabsorbed a bunch of ions as well. So you've reabsorbed stuff that you can recycle, stuff that you can use, stuff that's useful. And so now by the time you get up to your distal convoluted tubule, the main concentration that's in your filtrate is stuff that's like urea and potassium and protons and stuff that you don't mind getting rid of anyways. That's the whole idea here. And then, if you have a very specific hormone, do you guys happen to know what hormone allows you to reabsorb water in the collecting duct to then reconcentrate your urine? In fact, you only have concentrated urine if this hormone is present. Otherwise, you're not able to reabsorb water. It's a really important one. And its release gets blocked by caffeine. Good call, ADH, otherwise known as vasopressin. So if you drink coffee, alcohol does the same thing. It blocks your body's ability to release ADH, which means you can no longer reabsorb water in your collecting duct, which means you're going to be peeing out a lot of fluid, and it's mostly water, very clear urine, which is thus going to dehydrate you. That's why they say drinking coffee actually dehydrates you, because it stops your ability to actually reabsorb water from your filtrate. It can be problematic, but sometimes coffee's worth it. It's all balance. All right, so here's our last thing here. So basically, if we have water, aldosterone's what does the salt. We cover that in other channel classes. But what we mostly want to show you here is how brilliantly this system is designed. Because notice, this is your concentration. As we said before, it's really concentrated down in the belly of the nephron. Whereas in the top, it's about your standard blood concentration. So as you start to get your fluid to move through the nephron, we've got this other system that flows in the opposite direction. So this is actually a blood vessel known as the vasa recta. You can see it labeled there in the lower left-hand corner. So the vasa recta actually flows in the opposite direction of your filtrate. This is very useful because remember, as we said before, you're actively reabsorbing salt from your filtrate in the thick ascending loop. So as that sodium gets pulled into the vasa recta, it's then going to move down in the other direction, just in time to get to this side of the nephron where that extra sodium will draw water out. This is the countercurrent multiplier system. The idea is that you get to reabsorb sodium, and that active process of reabsorbing sodium is going to thus passively reabsorb water, both of which, as you know, we desperately need to survive. Pretty cool, right? I love this animation. It's like, oh, I see how sodium leaves this side, comes around this side, reabsorbs the water, because remember, this is the stuff that you definitely want. Cool. Oh, yeah, and so as Nicole's saying there, oh, no, never mind, Archway, I got you, perfect. Cool. All right, guys, well, that's actually the last slide that we have here. It's been so much fun working with you. Hopefully you've learned a bunch of things about the kidney. I'm confident you probably have more to learn if this is your first time. This is just an introduction. But you gotta make sure whenever you're studying for the MCAT, make sure that you are applying it, keep practicing, not just studying. So work on practice questions, work on practice exams, as Aishwarya brought up earlier. That's a big thing. And as we said before, really try to connect the dots between everything. Make sure that you see how the context relates. All right, so as the sun is setting, I will hop off and give Aishwarya time to jump back on camera, but it's been great working with you all, and I will hope to see you all in the future on the MCAT channel.
Hello, hello, hello. Welcome back, everyone. Like Tiz said, oh, John is fabulous. And it's so weird because I spent so much time watching John as a student. And so it's really neat being on this side of it, being able to learn from him, become a better teacher, things like that. But I assure you, John is one of our best. He's an amazing teacher. And like John and Tim were saying, we have classes like that every night. So I'm going to jump us over to my MCAT channel slide so you guys can learn a little bit more about that. And I'm going to talk about it a bit more too, because it's honestly one of the best parts. It was one of my favorite parts of the course. So just a little preview for those of you guys who are curious. Some of the MCAT channel sessions that we have coming up through the course for Monday include neuroanatomy, oxidation reduction, reactions of organic chemistry. Some other things coming up this week are acids and bases pH problems, hemoglobin, blood typing, resistors. So there is a lot of information. There's a lot of classes or channel sessions available to you for that more targeted prep in your weak spots. And Nicole, that's a great question. I'm glad you asked. So within our Kaplan course, you attend 12 sessions. And so those core sessions help you go over strategies for approaching the MCAT. You learn high yield science topics. You put those strategies into play by going over passages and learning how to do things like outline and predict. And it's all in the context of the MCAT. So everything you're doing is MCAT style problems and things like that. But you also recognize at Kaplan that everyone has a different background. Like I personally, I was a chemical engineering and bio major. So like bio, physics, that was not electricity and magnetism, those were kind of my strengths. And so I knew going into prepping that I didn't want to be spending a lot of time doing review over those areas because that wasn't really a good use of my time. And so similarly to that, like let me know, what were your guys' majors? What are the areas that you guys think you're really, really strong in? Because one of our goals here is that you spend your time as effectively as possible. And so what that means is that you don't spend a lot of time reviewing what you already know. You spend that time reviewing the things that are going to give you the most point gains. And so the channel sessions are optional live sessions that you attend in addition to your core classes. And again, they're over topics like the ones I mentioned. So those are really high yield topics, topics that appear very frequently on the MCAT. And you go to the ones that you need help in. You go to the ones that are kind of in your weaker areas. Awesome. I see some nursing, biochem, public health, bio. Totally. French, even French is helpful, like John was saying, because you have those root words that you can use to help you figure stuff out. So again, all of you guys have different backgrounds, different strengths, things like that. And that's something to keep in mind as you prep, that you want to find the option that's going to work best for you. And so one last thing about the channel, again, we also know that just because you guys might need to review these topics doesn't mean that your schedule will necessarily coincide with the MCAT channel. So what's great about it is that if you can't make one of the live sessions, or maybe if you just want to review something that you already saw, you can go and rewatch that session. You can watch a recorded version of it, and so that you have access to 24/7. So if you're up at like 4 in the morning and you're just like, I'm really feeling kidneys today, you can go and watch that kidney session. You can get that kidney review in. And so that's really fabulous because, again, you're able to target the stuff that you're kind of weaker on while spending your time in a way that's giving you the most yield. So are you guys with me so far? Are you guys pumped to keep going to learn more? Because I am. I'm really excited. So I'm going to take us over to some best practices. Because like John was saying, one of the things you want to be doing is making sure that you're doing practice problems as you prep. Because it's not just about sitting in a library and studying. It's not just about learning the content, because that'll get you somewhere. It'll get you, maybe halfway there, a third of the way there, but it's not going to get you all the way there. And what's really going to get you all the way there is making sure that whatever you're learning, you're actually able to apply. And so that's when, you want to be sitting down and doing practice problems after every content review session. You want to be doing at least 5 practice tests before your test day. And again, I'm going to break that down because I have a lot of students who are like, why 5? And they'll come to me after practice test 1 or 2 and they'll say, Aishwarya, my scores haven't changed. I'm exactly where I am at the diagnostic. I'll be like, yeah, that happens. That's very, very common. Because you go to class and you learn these strategies, you learn these skills, you learn how to outline, how to predict, you understand how the test makers structure their wrong answer choices so they become easier to spot. But it takes some time to learn how to do it. So I'm a swimmer. I am also a person who at age 5 refused to go in the water. And basically, my mother was like, we're in Florida, we're surrounded by water, you have to learn how to swim. And so she enrolled me in the swimming class and they taught me, okay, here's how you doggy paddle. Here's how you put your face in the water and don't die. And it was one of those things where I didn't just like enter the water and magically know how to swim, nor was I very good at it. I looked kind of like a floppy puppy just splashing around in the water. But then fast forward 15 years later and I was on a swim team because I loved swimming. And so I wasn't able to develop my swim team level skills overnight. And that's kind of like how the first and second practice tests are. Those are those tests where you've just learned the skills. You're still perfecting them. You're still the like floppy puppy splashing around in the water. But by the time test 3 and test 4 and test 5 roll around, you've built up some of your stamina for that 7-hour test. You have become better at outlining. You've become better at predicting. And you're just all around more confident. And that's why you want to make sure that you are scheduling in time for those extra practice tests. And like Tim said, we have 15 practice tests available in Kaplan. You have plenty of options, and those tests also include the AAMC practice tests. And I'm going to add on to what Tim was saying and saying one of the great things about the courses that we offer is that, again, in addition to our Kaplan resources, you get access to every AAMC resource that's been released. So you have your AAMC practice tests, and you also have AAMC practice sessions, sections. And so those are basically hundreds of questions on each subject that you can do for that extra targeted review if you know that you are weak in one of those sections. And so that's really, really great to have access to. And again, if you're not sure what is right for you, if you're still confused about where you want to begin your prep, you can give us a call. You can schedule an appointment. You'll see that there's another link that's pinned to the top of the screen right above that raffle link. And you can use that link to schedule an appointment. And again, what's super cool about scheduling an appointment is that you get access to an actual Kaplan MCAT teacher. So you get access to one of us who, we've worked with hundreds of students. We've seen pretty much every kind of area of growth that's possible. So we really know what kind of resources are going to be most beneficial to you, and we can steer you in that direction. So if you're not sure, you can call 1-800-CAP-TEST, or you can use that link at the top to schedule an appointment. And Nicole, and like Tim is saying, we have both the AAMC test and we have our Kaplan test. We recommend that you use both. Usually we say you want to save the AAMC ones at the end, until the end of your prep, just so that you get that AAMC practice right before you take your AAMC made test. And again, you have access to all of your tests. You have access to the question bank. So that's perfect if you have just studied the kidneys and you want to now test your ability to answer kidney questions, because you can choose to be presented with questions that are only kidney based. So one thing I'm going to move us to, because I think this is something that's really important to consider as you're prepping, is what type of class is ideal for you, what fits your schedule, what fits the way that you learn, and where are you going to get the most benefit from. So Kaplan offers a few different options. And what you've been experiencing today, what you saw with John, was an example of our live online setting. And so basically you have one teacher on camera, they're teaching the content to you, but you also have the ability to message in and talk to off-camera teachers. So some of the off-camera teachers you guys have been chatting with today include Brandon, Timothy, and Shelby. And so we have some really, really great people, Mary, Rohit. And what's cool, especially for those of you guys who are here right now, is that a lot of them have been medical students or are currently medical students. So for example, Rohit is about to go into medical school starting in August, whereas Mary is a med school graduate. So it's really, really great for you to ask your questions here to get that information that you want, but also use that link at the top to schedule an appointment and get personalized feedback on what is the best option for you. So in person is very similar in that you have your instructor speaking with you, but it's a little bit different because you are in a physical classroom. You are surrounded by your classmates. And so if you're the type of person who falls asleep really, really easily if you're sitting in front of a computer and you need kind of that watchful eye of your teacher staring you down to stay focused, then that's a pretty good option for you. That's one that you know will keep you alert, keep you engaged. So it's definitely something to consider as you're trying to figure out what types of prep are going to get you to where you want to be. Because at the end of the day, when you think about the MCAT, it's the first step in your med journey, right? So you take the MCAT, you get your score, you apply to med school, you get into med school, and you go from there. And so it's such a big investment that you're making in yourself right now. You're going to be spending so much time, you're going to be putting so much of your emotion into this, you're going to be putting a lot of money into this. So this is the start of that investment to get you where you want to be. And so it's very important to make sure that you are putting everything you can into this, that you are getting that score that you want, that you are getting the resources you need to learn the things that are going to boost you that extra 10, 15, 20 percentiles. Because at the end of the day, your MCAT score is basically the biggest thing that's going to get you that interview invite for the med school that you're interested in. So again, make sure that you're taking this seriously. You're thoroughly looking at what you have, what you need, what your weaknesses and strengths are. And so for those of you guys who took a practice test yesterday or today, give me a shout out in the chat, give me a woot woot. And I'm also super proud of you guys who are still here, who are still being engaged, who are still excited. For those of you guys who haven't, definitely go ahead and sign up for one. We have one coming up on February 1st. We have one coming up on February 5th. It's a great place to start. It's a great place to figure out what resources are going to get you where you want to be. And we're also, this is our final call for the drawing. So if you haven't used that second link, go ahead, fill it out, enter for that $50 Starbucks gift card, and we'll be drawing the lucky winners soon. Jesse, that's totally fine. I'm all for woot woots. But I do recommend that if you haven't taken a practice test, go ahead and sign up. It's a great thing. It's a great way to start your prep. And it's also a good way to realize that the MCAT is less scary than you think it is. Because I have a lot of students who say, I'm going to wait until I have gone through every content review book, until I have gone through all of my textbooks before I take a practice test or do a single practice problem. And guys, oh, please don't do that. That is by far the worst thing you can do. So, okay, promise me right now that you guys will be doing practice problems as you prep the whole time through. Yeah, Tim's right. You want to practice right away. So give me a 6 in the chat. Give me that promise that practice as you mean to continue. Exactly. Great. And so I'm also going to say that we are running a pretty cool promotion. So I saw from one of the first polls that I had you guys fill out, the when are you registering to take the MCAT poll, that a lot of you guys are signing up for, or a lot of you guys haven't registered yet. And that's totally cool. And for those of you who aren't sure, the registration for July, August, and September dates actually opens on February 7th. So definitely put that on your calendar because test dates fill up real fast. So you want to get on those. But for those of you guys who are prepping in those times, we're running a promotion right now. It's actually really cool. I've never seen this promotion on Kaplan before, and it's one of the best we've ever had. And it's called Unlimited Prep. So basically, if you sign up between now and Wednesday, January 31st, you get access to your MCAT course until September 19th, and you also get up to $300 off. And so again, it's one of those things that it's probably the best promotion that you will have access to, and it's amazing because you get access to your resources, the MCAT channel, all of the practice tests until the end of the testing season, so until mid-September. And so we're going to throw that link into the chat for those of you who are interested. We're going to throw that code in there. And so definitely take full advantage of that. And Hanan, I see that you asked how early do you recommend starting prep? And I would say most students start prepping about 3 months before their MCAT test date. And that's because our courses run for 6 weeks. And so you take your course for 6 weeks and you learn your skills, your strategies, you do your content, and then you want to give yourself at least 4 or 5 weeks to be able to take a practice test every week. And I don't recommend doing more than that just because they are fatiguing, they are very tiring, and because taking those practice tests is going to guide how you prep the rest of the week. It's going to guide what you pay attention, or focus what you pay attention to, and things like that. And so again, that's also limited by what your schedule is like. So if you know that maybe you're going to need longer to prep just because you have such a busy schedule, then it's definitely good to keep that in mind. We also offer some classes that are once a week for people with busier schedules. So again, if you have questions on that, if you want to get a better idea of when is a good time for you to start prepping based on your personal schedule, use that link at the top and schedule a free meeting, because again, your teacher that you talk to is going to have a much better ability to tell you when to start based on your situation. All right, so what I'm going to do now is open it up to questions. Oh, awesome, Jesse. When should our last full-length practice test be before the test date? So I would say you want to keep doing those practice tests until the week before. So definitely the day before your test, you don't want to do anything. Like, you want to basically be sitting on your couch, watching Finding Nemo, and eating chocolate, which is totally not what I did at all. But you want to be chilling out for that day before your test. But if you take your last full length a week before, then again, I 100% recommend that it be an AAMC one, just because that will be your last exposure to what those questions are like before you take the actual one. But that's a pretty good way to plan it. What other questions do you guys have for me? No, Jordan, I do not. Because at that point, you've learned everything that you're going to learn. You've studied your content, you've practiced your skills, you are good to go. So I recommend the day before the test, that's your Zen day. It's not going to improve your score. There's nothing you're going to learn in that day that's going to change very much. I mean, the one thing I would say, if you really want to, I was a big fan of writing out all the equations that I would need for the exam for about 2 weeks beforehand. So I did do that right before I went to bed, just kind of like because it had become a habit for me, but I do not recommend, doing heavy content studying or things like that. Laura, you're going to probably keep doing some content review up until the last day you study, just because, sometimes you forget random details and you want to go back and look in your book. But most of the first roughly 6 weeks of your studying, if you're doing the 3-month plan, are going to be your heavy content review period. And then after those 6 weeks, you're going to transition to more practice-based content review. Does that make sense? So Joy, my best advice to students retaking the MCAT. So if you retake the MCAT, you want to make sure that you are going to be increasing your score from the last time heavily. So my best advice to you is take practice tests, because you're gonna, by the time your practice test scores start stabilizing, you know that that's what your score is going to be on the actual MCAT. So if your score is stabilizing and it's not stabilizing where you want it to be, then that's probably an indication to you that, one, you might want to push back your test date, and two, you might want to look into some new resources, or you might want to take advantage of resources you haven't been taking advantage of. Because again, if you're going to retake the test, you need to show medical schools that you retook that test for a reason, that your score increased so much that you were able to learn whatever you didn't grasp the first time. Nicole, if we do the in-person route, we get the online med channel and all of the benefits from the past slide. Yep. Here, I'll actually take us to this slide over here. So if you take an in-person class or a live online class, you get access to pre-recorded lessons in addition to the in-person lessons that you'll be attending. You get the QBank, the AMC resources, the MCAT channel, all of that stuff. And yes, Maddie, you get quick sheets, and those have all the equations that you need. I personally recommend making a mark next to the equations you use when you're reviewing a practice test, because that gives you a better idea of which ones are really, really common. So that way, you get more and more used to that. Laura, if you're doing the 3-month plan, you actually take your first practice test after your 8th class in the course. And so since classes usually run 2 times a week, that's about 4 weeks in. Do I suggest taking the full length or diagnostic before getting fully? Oh my gosh, Patty, yes. I 100% recommend taking a full length or diagnostic before doing content review, because one of the things you're going to find is that the stuff you think you need to review is probably not what you actually need to review. And so taking that full length or diagnostic shows you where your actual content gaps are versus where your maybe strategic gaps are. So like, definitely go through an exam and check, did I miss this question because I didn't actually know the content, or because I was moving too quickly, or I just didn't understand how electricity played into neurons, and I wasn't expecting an electricity question in this neuron passage. So that's definitely something you want to do before you dive into content review, 100%. What questions have I missed? All right, cool. So if you guys have more questions, feel free to keep posting them in. Meanwhile, we're going to do a drum roll for the drawing. Sorry, my computer doesn't really, it's not a very good drum. So while we wait for that, I want you guys to shake it out, to do a little drum roll on your own computers. Maddie, are the quick sheets given in the Kaplan book set? Yep. So your Kaplan book set comes with a content review book for each section. It comes with your lesson book, and it comes with your quick sheets. All right. So Alyssa Wallowin, I apologize if I mispronounced your last name. You have been chosen for the $50 Starbucks gift card, and we will be reaching out to you via email. So keep an eye out for that email. For those of you guys who were not selected this time, we have some really great other free prep week events coming up. If you like today's session, we have acids and bases tomorrow, and we have molecular genetics after that, and we're rounding it out with an MCAT boot camp on Wednesday, which is great. MCAT boot camp is where we go through some of our really challenging questions, and we show how using the strategies that we teach in class can really help you break down those questions and make them a lot easier to approach. So come back each night for some more great study strategies. Come back for another chance to win. We have some really great raffles coming up, and definitely use that link at the top and schedule a meeting. It's the best thing you can do if you're not sure where to prep, if you don't know what resources are best for you. And they're definitely here to make sure that you are going to come up with a winning plan for the score, for the goals that you have in mind. It's definitely worth it. Med school it's a whirlwind, it's a roller coaster, but it's also a lot of fun. And it's really important that you invest in yourself, that you get the resources you need, because I promise you, everyone can get the score they want. It's a matter of putting in the time, but also using your time wisely. And I want you guys to be excited. I want you guys to know that med school is like, it's incredible. You learn these amazing things. Like we covered kidneys today, and I know that we were talking about kidney physiology, but the amazing thing is the foundation you're building now, it plays such a huge role. So we personally spent about a month and a half going over kidney diseases and disorders in my school. And it's incredible how we've built on all of the stuff that John was talking about today. So definitely invest in yourself. Definitely put in the effort now to learn this stuff and understand the concepts, because this is what you're going to be building on in med school. And I know you can do it. I know you have it in you. And again, it's really, it's all about helping you guys fulfill your dreams. It's all about getting you where you want to be. So keep asking us questions, keep messaging in. We're here to answer, to help, to give you advice. I am going to hop off camera, but I will still be here. And definitely come visit us tomorrow. Come learn about acids and bases. Come see me and some really awesome teachers again. And so with that, I'm going to leave you. You guys have been amazing. Again, for those of you who were with me for the practice test and this, I am so proud of you for working through it, for staying focused. For the rest of you, again, congratulations on investing in yourself, on deciding that you are going to study and you're going to put your all into this. And congratulations for coming here today. It's definitely a sacrifice, but it's well worth it. All right, have a great night, and I will hopefully see you guys soon.